my honor and privilege to introduce our speaker for the 2024 Helz Lecture. This is the third Helz Lecture that has been held. The first was by Gene Likens and Sue Brantley, and today our speaker is Andrew H. Knoll. He is the Fisher Professor of Natural History at Harvard and recent recipient of the Crawford Prize of the Swedish Academy of Sciences. This is the highest honor that a geoscientist can get, so it's very important. Knoll's research focuses on the early evolution of life, or its environment, our environmental history, and especially the interconnections between the two. For the past decade or more, he has also been a part of the Mars rover teams. I show some pictures here that I particularly like of Andy. Here he is talking about Mars. He has two watches on because he's monitoring both Mars and Earth time. And he's looking at some blueberries there. This is a picture I took of him with a 3D camera. I love it particularly because of the polar bear in the background, and he has a nice story about that. So here's Andy being awarded the International Prize for Biology in 2018 with Emperor Akihito and Empress Michiko, and more recently the Crawford Prize, the highest award that a geoscientist could get with Crown Princess Victoria. Other honors that Andy has received include the Walcott Medal and the Mary Clark Thompson Award, Medal of the National Academy of Sciences, the Phi Beta Kappa Book Award in Science for his book on life on a younger planet. The Moore Medal for the Society of Sedimentary Geology, the Paleontological Society Medal, and the Wallace Medal of the Geological Society of London. He's a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the American Philosophical Society, and the American Academy of Microbiology. He received a BA in Geology from Lehigh University and his PhD in Geology from Harvard University with Elsa Bardorn. Andy is a world traveler. These are a few of the trips that I've taken with him. Here we are in South Australia, his colleague Malcolm Walter. Here we are looking for elephants. We found quite a few that day in northern Namibia and Tosha Park. And this is a picture, I didn't take this, but this is him up in Svalbard looking at one of the glacial deposits that we have been studying for many years. Andy was a member of my PhD committee along with John Hayes and Case Klein. One of his premier students is actually here, and he's being inducted into the National Academy, Shuhai Shao. Andy, of course, rubbed elbows with Stephen Jay Gould, and this is a picture of an early group event at one of the Geological Society of America meetings. Andy's academic shadow is wider and likely more visible than the solar eclipse next week, with proteges spread across the globe, some of whom are joining us virtually today through live streaming, and I welcome all of them. I'm grateful for being part of this community over the years and for the scientific path that Andy's perceptive vision of space and time has revealed to us all. Before I let Andy come up, I also want to thank once again George and Roz Helz for making this lecture series possible. So, without further ado, we will start Andy's lecture on the long conversation between Earth and life. Andy Knoll.
Okay, well, thank you, Jay, very much for that kind introduction. I'm glad to be here. It turns out that this is not the first time I've been to College Park. When I was a student, which is sort of back in what geologists call the Holocene, the place to be in the fall, if you're interested in the origin and early evolution of life, was the University of Maryland, because there was a prominent student of chemical evolution named Cyril Panaparuma who put on a meeting called Chemical Evolution and the Origin of Life. And this was before NASA did anything and other institutions. And so it was a place where even as a graduate student, I would get to meet people like Stanley Miller and Leslie Orgel and this. It was all very exciting. So I have this sort of warm memory of life with Cyril and the meetings he put together. Now, what I'm going to talk about, if I can get this to move forward, sorry. Maybe next as well. Oh, there you go. Everyone knows that fossils record the history of life. And if you know one thing about fossils, you know there were dinosaurs long ago. They were big, they were mean, they were really something. And I agree with that. I think dinosaurs are really wonderful, but they were around for something like 5% of the history of life. Now, if you know a little bit more about the fossil record, you might know that the organisms you see to the right of the dinosaur skeleton, called ammonites, were major constituents of marine biotas at the time that dinosaurs ruled the land. And if you know even a little bit more, you'll know that long before there were dinosaurs and ammonites, there were animals on the seafloor, like the trilobite you see below you. So for many people, the fossil record, even in a somewhat extended realm beyond dinosaurs, is a record of animals, basically a record of us. So I think it's useful to put that into perspective, and I'd like to do that with three images. The first one I really like. This is it's from The Economist, but it is a rendering of information in a paper by my friend Ron Milo in Israel and his colleagues from a few years ago, where they attempted to quantify the biomass of all organisms on Earth. And what you see there, first of all, is that plants dominate the planet's biomass. Now, plants cheat, it turns out, because if you go and examine a tree outside the building here, you'll find that more than 90% of it, the wood, is not living tissues. So if you only take living tissues of a plant, then bacteria win. But no matter how you slice it, animals are that tiny little red rectangle in the top. Animals are a very small part of the biomass of this planet, and actually a limited part, probably, of the diversity as well. Now, to put that in some kind of relative time frame, I'll show you this diagram on the lower left, which is what is sometimes called a universal phylogeny, and that's a hypothesis of the evolutionary relationship among all organisms, based largely on sequence comparisons of nucleic acids and proteins. And while this kind of diagram can get you into a bar fight at a biological meeting, because there's lots of debate about individual phylogenetic relationships among taxa, there are two things in this diagram that everyone agrees on. First is that life can be divided into a very small number of major domains, and that at the beginning, the immediate descendants of the last universal common ancestor, you had two groups, both of which are what have often been called prokaryotic, the bacteria and their sister group, only identified as distinct from bacteria in 1977, called the archaea. And then the third major branch, the eukaryotes, organisms with nuclei and things like this, is actually a hybrid in the sense that a good portion of the eukaryotic biology does seem to be derived from a group of archaea only discovered in 2015, and part of it also comes from bacteria. And when you look at the phylogeny of eukaryotes, again, what you see is that animals, all of the animals that occupy the last 600 million years of the fossil record, are one small branch at the distal tip of the eukaryotic tree. So that tells us both that there's an immense diversity of life on this planet, mostly microbial, and that there is, by implication of this diagram, a much deeper history of life than that represented by animals, and that is also microbial. And then if you look at this timeline over on the right, the oldest direct evidence we have of animals as fossils is about 575 million years ago. We'll talk about that a little bit. But of course, our planet's about 4.6 billion years ago. And the question is, how much of this antecedent pattern of evolution that we infer from phylogenies can be captured as a historical record in sedimentary rocks? And so that's what we're going to do today. Look through this area in here, the long history of life actually recorded by sedimentary rocks older than those that contain animals, and see what it tells us. Okay, we're going to start in some ways where I started when I was a young scientist, and that is in the glaciated highlands of Spitsbergen, which is an island about halfway between the northern tip of Norway and the North Pole. And this is a remarkable place. It's kind of the Alps at sea level. But as you can see here, rocks that antedate the oldest record of animals are beautifully preserved, beautifully outcropping. And so we can begin to ask the question, if there is an older record of life, largely microbial life, will microbes, tiny, transient beings, leave a tractable record? Well, I probably wouldn't be giving this talk if the answer were yes, but I think what I want to emphasize here is that that column of rocks that I just showed you is very much a chapter in the history of our planet. We live on a planet that records its own history, and one of the things that we can see just in this picture here on the upper right is that the textures of rocks, their physical properties, tell us something about life and environment. So in the upper right there, that's a carbonate rock, like a limestone today, and you'll see there are these kind of wavy laminations. And if you want to find those kind of laminations forming in limestones today, you go to places like the Bahamas, where mats of microorganisms spread out on the sediment surface, and then they get covered, and you get these textures imparted to the sediments. Also, if you look at the top, there's this place that it looks sort of like a teepee, and geologists call these teepee structures. And what they represent is places in the upper part of the tidal zone where there's a lot of evaporation during low tide, and that causes carbonate minerals to precipitate within the sediments, and the pressure builds up, and it just bends the rocks upward. And so this rock, only by its physical features, tells us that there was microbial life living in the most exposed parts of tidal flats 750 million years ago. Now, it turns out if you take a sample of this rock and cut it down to paper thickness and look at it under a microscope, if you do that in the gray carbonates, all you'll find is interlocking crystals of carbonate minerals. Not very edifying from a biological point of view. But if you look at these black structures here, which are concretions of silica, SiO2, chert, or flint, these formed in the sediment soon after it was deposited, and they act like bioplastic in that they are able to preserve over intervals of billions of years textures, including biological textures, that were present in the original sediment. So what you see below that, this is just a photographic image of a part of that section, and these are cells. Each of these cells is about 4 microns in diameter. We think they are cyanobacteria, which are photosynthetic bacteria that all the textbooks will tell you have green plant photosynthesis, but if you've studied biology, you know that green plants have cyanobacterial photosynthesis because the chloroplast of the tree outside here is actually descended from a once free-living cyanobacterium that was engulfed and reduced to metabolic slavery. So we already have at least a first-order answer to our question. Yes, microbes leave a record in older rocks, and if we're diligent, we might be able to learn something from it. Here's just one population to give you a sense of what we can learn under good circumstances. So this is a kind of fossil that's fairly common in tidal flat assemblages from rocks of this age. This would be about 50 microns across. There was a cell in here. All that's left is this amorphous goo, but the cell was actually covered by a series of envelopes that it secreted through its lifetime. They're elongated in the downward direction, and that formed a stalk that kept this cell in place relative to movable sediments in which it was living. You sometimes find these as individuals. You often find them where a large population is covering a bedding plane. And from the fossils alone, we can reconstruct a life cycle for this organism. And it turns out that, and this is very good for someone like me who used to go to the Arctic in the summer, that if you want to go and look at a modern equivalent, you go to the Bahamas. Good place to go in the winter. And so here we are working in the Bahamas, and on the right is just a surface in the tidal zone, and those black pustules that you see turn out to be populations of these cyanobacteria. And so this is in an environment thought to be very similar to the 750 million year old environment represented by those rocks I was just talking about. You have microorganisms whose morphology and life cycle and distribution is very similar to what those ancient fossils found. So it gives us some confidence that when we're looking at those microfossils, we are looking at cyanobacteria whose biological properties were similar to things we can see today. And it turns out that if you look in different environments represented by different kinds of sedimentary rocks, there's a diversity of fossils. There was ecology 750 million years ago. So there's a variety of things that we think are cyanobacteria, but there are also many different eukaryotes. So you see that vase-shaped fossil, which is probably made by something called an amoebozoan, a distant relative of humans. That's about 100 microns. There are things that are large, like this cell with a cyst inside it. That's about 0.5 millimeter across. We don't know phylogenetically who made it, but it was certainly a eukaryote. There are things that are very similar to certain kinds of green algae today. So in these rocks, 200 million years before the oldest fossil animals, we find evidence of fairly diverse microbial life. And that's not all. In fact, the most conspicuous evidence of life in these rocks are something called stromatolites. Now, we know a lot about stromatolites because there are some places in the world where they form today. This is a place called Shark Bay in Western Australia. I think for people who study the Precambrian, it's sort of like the mecca of these studies because we can actually do experimental and observational work on these and find the relationship between microbial communities which spread across a surface, trap and bind sediments, and precipitate carbonate cements. And this happens through time. You build up layers and you get structures like this, these layers. And these things can be columnar, they can be domal, they can be conical. And if you look in the upper left, in the middle of that cliff, there are these kind of tan lozenge-shaped structures, which are 4 or 5 meters thick. Those are reefs. Those are patch reefs built by microbial communities. So the stromatolite record, independent of any microfossil record, tells us that the seafloor in the photic zone of ancient oceans was covered with microbial communities, some of which built these rather remarkable structures. And then there are chemical clues to life. One of the things that can be found here and in other places, and I'll talk about this in a bit of detail later on, are chemical biomarker fossils. Now, it would be lovely if DNA or proteins could be preserved But turns out DNA and proteins are really good to eat, and so they usually disappear soon after the death of an organism. I think the oldest unambiguous fossil DNA is about 2 million years old. But what preserves well are sterols. Now, for years I would tell my students that when they die, the last part of them to be contemplated by their descendants will be their cholesterol. So something to think about. At the top, that's the structure of a sterol or a sterane, the geologically derived product from a sterol. And those preserve their lipids, and they preserve very well. The bottom one is something called a hopane, and that's a structure made by different kinds of bacteria that also preserves well. So they provide evidence, in many cases, of organisms that have not left a morphological fossil record, so they expand our sense of the world. And then there are geochemical records, and a couple of people in this audience, Jake Hoffman, James Farquhar, and some others, study these in detail. Microorganisms may be tiny, but they're so abundant that they can actually affect the chemical composition of seawater. So one of the things that actually when Jay was a student, we did together was to look at the carbonate carbon and organic carbon isotopes, C13 and C12, of those Spitzbergen rocks. And what you find if you go to the Bahamas today, that the ratio of C13 to C12 in the organic matter buried in these carbonate sediments today is more enriched in C12 by about 25 parts per thousand than the carbonates that it co-occurs with. And the reason for that is that when photosynthetic organisms take CO2 from their environment and fix it into organic matter, there is a preference for the lighter isotope C12. And so that's what we find 700 to 800 million years ago, and as you'll see, that's pretty much what we're going to find for the entire recorded history of life. So it's a very strong signature that there was a vigorous carbon cycle at this time. If I showed you data on sulfur, I think I could conclude persuasively that there was a vigorous sulfur cycle at this time. There was a microbial nitrogen cycle. So you have complex microbial communities. Okay, so here's point number one while you look at some modern microbial mats in the Turks and Caicos Islands, that if you go to places where you have microbial communities today and study them, they provide keys to understanding the kinds of fossils, sedimentary textures, and chemical parameters that we are seeing in the geologic record. And what they tell us is that, 700 to 800 million years ago, there are no animals, but the fingerprints of life are everywhere. So what happens if we dig deeper? Okay, this is a place that I enjoyed working when I was younger and fitter than I am now. This is in northern Siberia, a place called the Great Wall, because on the other side, if you took a picture of this from half a mile in the other direction, you'd see exactly the same thing. It's just this big sliver of rocks. These rocks were deposited about 1,500 million years ago, and they're beautifully preserved. And once again, there are fossils of cyanobacteria, that short filament that you see, it's a 10 micron scale. There are eukaryotic cells. So again, already we have eukaryotes. I love this picture on the left, largely because the person standing there, my colleague, the late Misha Semakatov, was exactly 2 meters tall when he had a hat on. So he's a very good person to use to make photographs. And you might say to yourself, well, I see what you're saying. There's that viscid-shaped thing, that's a microbial reef. And that's right. If you knew that, 5 points. It turns out it's a 15-point question. It turns out if that's, a reef maybe a little bit bigger than this table, note that Misha is standing on a rounded surface, which is a microbial reef that would be somewhat bigger than this room. And the wall behind him, you can see if you look carefully, there are these vertical structures which are stromatolites. That's a microbial reef larger than this building. So before there were animals, there were microbes that built reefs with the same kind of dimensions that we associate with coral reefs today. And in fact, for most of Earth history, these stromatolites are the predominant sedimentary features of carbonate rocks. So let's test our luck and double it again. And what you're looking at now is one of two places in the world where we have the oldest little metamorphosed sedimentary rocks that we know of. You know, sort of our earliest relatively clear window on life and environments. This is in Western Australia. And if you look where the people are concentrated, that sort of dark brown layer that you can see going up the hill turns out to be made of stromatolites. And we've done some work with our Australian colleagues that show at the microscopic level, it's very clear that microbial mats were involved in the formation of these structures. So there's evidence of life there. And then it gets a little trickier. It turns out that if you look at carbonate rocks and organic rocks, or organic matter in those rocks, that again you see the difference between the carbon isotopic composition of the carbonates and the organics. In this case, it's about 28 per mil, something like that. Again, that's telling us there's a vigorous biological carbon cycle. It's a little trickier for sulfur for various reasons, but I think many people have concluded there was a sulfur cycle as well. So life is there. Unfortunately, these rocks have been cooked enough that there is not a well-preserved record of biomolecules. And the record, people, every year somebody describes a new set of fossils from these 3.5 billion year old rocks. And they all tend to be these kind of 10 to 20 micron organic spheroids. And the picture you're looking at here is 10 to 20 micron organic spheroids, but it's actually not from 3.5 billion year old rocks. It's from much younger rocks where hydrothermal fluids have percolated through sediments. They've remobilized organic matter, and then it precipitates out in these spherical forms. Turns out that all of these 3.5 billion year old rocks have been subjected to hydrothermal fluid flow. So I think we have to be very careful about microfossils. So, second point. Available evidence suggests that life originated early in the history of the Earth. There are some metamorphosed rocks that take us back to about 3.8 or 3.9 billion years old that certainly have carbon isotopes that are consistent with the biological carbon cycle. And more or less, as we go back in time, we run out of rocks to look at before we run out of evidence for life. So life originated early on this planet, and what might make Earth distinct, if not only in our solar system, perhaps beyond, is not that life began here, but it's persisted for 4 billion years. That's not a given, so we must be remarkably lucky. Now, there's a lot of things we don't know and probably can't know about that early life, but there is one thing we're pretty confident of, and that early life existed in a world without oxygen gas. In fact, did so for more than 1 billion years. There's been a fairly large geochemical industry for decades now of people trying to understand the history of oxygen in the atmosphere and oceans. And one of the really important points that's emerged from the work of a lot of people is that about 2.4 to 2.2 billion years ago, oxygen begins to accumulate as a permanent presence in the atmosphere and surface oceans. Before that, there may have been little bursts of oxygen, but there was not a persistent oxygen-rich environment. There's a number of lines of evidence for that. One of the ones that people looked at early and still do, this is something called iron formation. Most of the iron that's used in industry comes from these early sedimentary rocks that are older than 2.4 billion years. And the reason that these formations form then, they can't form now because iron cannot be transported in solution through the world's oceans today, because when the reduced iron comes in contact with oxygen, it precipitates out as rust. And so in order to get these kind of rocks, you had to have basically anoxic or oxygen-poor seawater through which iron could be distributed until it was deposited. These kind of iron formations are fairly standard parts of sedimentary successions prior to 2.4 billion years ago. And then there's a couple of reprises, they are not common at all. Also, if you look in older sedimentary rocks, you find that there are minerals like pyrite, siderite, which is a iron carbonate, uraninite, a uranium oxide, which again are unstable in the presence of even 1% of today's atmospheric levels of oxygen. And we find that before 2.4 billion years ago, they accumulate in sedimentary rock. So this is a picture of some 2.7 billion year old sandstones from South Africa. And this clast or particle that you're looking at, the golden stuff is pyrite. This thing was weathered out of a rock face, carried by rivers down to the coast, deposited in a delta without ever coming into contact with much oxygen. After 2.4 to 2.2 billion years ago, you don't see this stuff anymore. And then there's a wonderful tool to which we owe to James Farquhar, a member of the faculty here, using what's called mass-independent fractionation of sulfur, which I won't go into in detail, but basically in a world without oxygen, or with oxygen in only extremely small quantities, there are what are thought to be photochemical reactions in the upper atmosphere that produce a particular type of isotopic signature, and that's present fairly commonly and consistently before 2.4 billion years ago, and then you don't see it much anymore. So there's a variety of lines of evidence that suggest that 2.4 billion years ago, the world is changing in a fundamental way, and fortunately, all the lines of evidence pretty much converge on a single time. And I talk about our hero because there's only really one source of oxygen that's capable of oxygenating the planet, and that's oxygenic photosynthesis carried out by cyanobacteria. Now, there's a lot of debate currently on what actually controls that GOE. Why is it that after more than 1 billion years of anaerobic life, we all of a sudden have this world that's fundamentally different and more oxic? And there's two lines of argument that go into it. There are some people who think, well, it's easy. You just evolve cyanobacteria for the first time 2.4 billion years ago. They produce all this oxygen and the world changes. But there's reason to believe, many people believe, and I'm one of them, that there's evidence suggesting that cyanobacteria existed for a reasonably long time before the GOE. And if that's true, then we have to look at environmental explanations for the oxygenation event. And two lines of evidence that might be used to support the idea that cyanobacteria are more ancient and only oxygenated the planet well after they first appeared are one, molecular clocks, that is, using fossil evidence to calibrate these phylogenies in terms of time, and almost all of those clocks suggest that cyanobacteria existed at least several hundred million years before the GOE. And then there are what are called whiffs of oxygen. The first whiff of oxygen paper is one that Jay Kaufman was part of. And that is evidence of oxygenated environments prior to the GOE. Not persistent, probably local and transient, but it's a geochemical record suggesting oxygen is around. Now, some people have argued that that oxygenation happened later, when later in the history of the Earth, oxygen-bearing fluids came through and altered the rocks. But there's something on the order of 30 of these, and if any one of them is correctly interpreted, there were sources of oxygen well before the GOE. A way of thinking about that that I like very much comes from the lab of Don Canfield in Denmark, that Jones et al. Paper. And what they argued is that if you look at the ratio of iron, which was a source of electrons for photosynthesis by groups other than cyanobacteria, which use water, and phosphorus, which is an important nutrient, that if the ratio of iron to phosphorus is high, so that you run out of phosphorus before you run out of oxygen, cyanobacteria will not be important. What that basically counts on is that in a world where there are other electron donors for photosynthesis besides water, that takes much less energy. And even today, where you have, anoxic sunlit waters where there are alternative electron donors for photosynthesis, cyanobacteria don't do that well. The other groups do. And so what they suggested was as long as you had, enough iron to outstrip the phosphorus supply, it's going to be pretty much an anaerobic world most of the time. But the minute you run out of iron before you run out of phosphorus, that's a world that's safe for cyanobacteria because they'll get their electrons from water. So that presents a very different way of looking at it. And then we can say, well, why might that ratio have been, what this implies that it was in the Archean, and how would it have changed? And one of the things that I've been fortunate to look at with a colleague who was trained just down the road here at Johns Hopkins now, working in China, Jihua Hao, and we, by which I mean Jihua, made some really nice models of nutrient availability in the Archean oceans based on what we're learning about continents. And that is, if you had flown around the world 3.5 billion years ago, you wouldn't have seen much in the way of continents. It was largely kind of a water world with, volcanic islands here and there, maybe a little bit of continental crust. But people are converging on this as being the way the world looked. Under those conditions, the amount of phosphorus that can be supplied by weathering and erosion is relatively limited. And then, as a very bright geochemist now at St. Andrews named Eva Stuyken has argued, that the Archean oceans were oceans that had very little in the way. It's not just that they didn't have oxygen, they didn't have other oxidants either. There wasn't much sulfate, there wasn't much nitrate. So under those conditions, the sort of respiratory remineralization of organic matter was probably very inefficient, as it is today in places like lakes that fit that description. And so therefore, that would limit the sort of upwelling return of phosphorus to the oceans. And to make it even worse, if there's iron in the deep waters, and there appears to have been, that iron would commonly react with phosphorus, or phosphorus could actually be absorbed onto iron. And so it all suggests that it may have been a very low productivity world. And this is just a image from the paper with Jihua, and it shows a number of people, for various reasons, have estimated how much primary production existed in Archean oceans. And even though people have different arguments for it, everybody seems to agree it's a low number. Maybe a couple percent of today's levels. What Jihua did was actually took several different models of continental emergence, and if you have late emergence, and I think probably if he did it again, he'd probably push it even further, that would have a big effect on nutrient supply. So just one more thing, and this is, I never thought as a paleontologist I'd be telling you about tectonics, but I think that we might essentially look to tectonics for a partial explanation of the Great Oxygenation event. Not by itself. I think one of the things you'll find, I believe that almost everything interesting that happened in the history of our planet happens when there's interactions between physical and biological processes. But the one thing we're pretty confident of is that the early Earth, the mantle temperature was higher than it is today. One of the consequences for that some people have talked about is that the mantle couldn't really support continents with high relief. It was just too weak because of its temperature. And also, my colleague Rebecca Fisher and her student J.J. Dong wrote a really interesting paper a year or two ago where they showed that at mantle temperatures that may have existed 4 billion years ago, the mantle can hold very little water. Certainly not the amount of water that exists there now. Now, there's probably at least an ocean's worth of water, maybe more, in the mantle. Their thought is that with the hot mantle, that all actually degassed out. So you might have had twice as much ocean water at the surface. And between those two events, then, there would be very little emergent material to weather and supply nutrients for life in the oceans. Okay, so what I would suggest then is that largely because of this sort of late Archean emergence of continents, which I think is very, very clear from the sedimentary record, that we really change both iron availability and nutrient availability in a way that by 2.4 billion years ago is favoring cyanobacteria as primary producers. There may well be other things going on, but I think that interplay between tectonics and biology has something to do with it. So if that happened, how much oxygen accumulated? Well, the answer is probably not much. This is now an almost iconic image from a paper by Tim Lyons and colleagues about a decade ago. And it is a cartoon in that it shows no change for periods of 2 billion years, and probably there's a lot more wiggling involved. But basically the idea that there was very little oxygen before the great oxygenation event. Then in Earth's middle age, which is most of the Proterozoic eon from 2.4 to about 600 million years ago, you have modest amounts of oxygen, maybe 1% or a couple percent of oxygen in the atmosphere and surface waters, and basically most deep ocean waters were oxygen free. And only toward the end of the Proterozoic and into the Paleozoic does oxygen climb to its modern levels and the deep ocean gets fully oxygenated. And there's a wide variety of geochemical evidence that has been put to play in this. There are different nuances with those different types of evidences, but again, I think they're largely uniform in telling us that for most of the Proterozoic eon, oxygen remained well below what we see today. And again, tectonics might have something to do with that. Yes, continents emerged, but, and there's a guy named Chris Spencer in Canada who's written some very, very interesting papers on this. There don't seem to have been high mountains. Again, perhaps because of a warmer mantle. We still have a low oxidant supply, so it's difficult to recycle phosphorus and all of that. So the world remains quite different from the world we know today. But in this world, there's a whole new type of cell, the eukaryotic cell. We actually have evidence for a modest diversity of eukaryotes beginning around, say, 1.7, maybe 1.8 billion years ago. You can see some of that evidence here, things that have large cells that have arms, that have spines, that have tessellated surfaces, that have sort of corduroy surfaces. These are things that are commonly associated with eukaryotic life, but not bacterial life. We don't know who made these. Phylogenetically, it's not easy to place them on a phylogenetic tree, but they do seem to be eukaryotic. And fairly early on in the middle of the Proterozoic, we find simple multicellularity. These two fossils here are probably our earliest evidence of groups of eukaryotic organisms that are still with us today. So this charming fellow comes from rocks that are a little over 1 billion years old in Arctic Canada, described by my former student Nick Butterfield when he was a student. Nick's now a professor at the University of Cambridge. And this one, which is 900 to 1 billion years old, described by Shuhai Xiao, my former student, here today. And that is, I think, correctly interpreted as a green alga. So by about 1 billion years ago, we're starting to see things that we recognize. But there's older evidence of macroscopic, not complex multicellular things, but sheets, blades, that kind of thing, suggesting that at least simple multicellularity had come to eukaryotes. In fact, this paper, which was just published a few months ago by Lanyan Miao et al. I'm an et al. In here, but Lanyan did all the important work. These are 1,635 million year old fossils. Were very well dated. Their size, these things are hundreds of microns in diameter. If this were discovered in rocks that are 200 million years old, it would just be called a green alga, and everybody would go home. That's maybe a bit too brave for this, but I think unambiguously these are simple multicellular eukaryotes telling us that multicellularity came to the eukaryotic clade early in its history, and I think they are very likely to have been photosynthetic. And what else is cool about these is that Lanyan actually found that some of these filaments have cells inside them. And these are, again, if you look at green algae today, certain lines of green algae make reproductive cysts inside their vegetative cells. So whatever the right explanation is, it suggests that we have complex life cycles already at this time. Now, so we get simple multicellularity more than 1,600 million years ago. It may be surprising, but we don't see evidence of complex multicellularity for another billion years. And when we start seeing it, it's really curious stuff. This is one of my favorite outcrops in the world. It's a place called Mistaken Point in eastern Newfoundland. It's well dated at 565 million years. And what's beautiful about this is you can see there are all these bedding planes exposed, and it turns out that they are just covered with fossils, largely covered by volcanic ash. This has been called an Ediacaran Pompeii, and that's actually not a bad description. So you have these macroscopic structures, and the question is, what are they? And it turns out, if you step back and say, well, let's ask how these things made their living. It turns out that let me just go back. There's no evidence that any of these things had a mouth or a digestive system. So they must have gotten their nutrition by either so-called phagocytosis, that is individual cells encompassing particles and then distributing their carbon content, or by what's called osmotrophy, where they take in organic molecules just the way fungi do. Also, there's no evidence that these had respiratory systems. There's no gills, anything like that. So again, we think that they exchanged gases by diffusion. And it turns out, if you say, well, are there any organisms that work like that today? This one does. This is something called a placozoan, a little bitty thing. It only has upper and lower cell layers called epithelia. It exchanges gases by diffusion. It moves by means of just cilia movement. There's no strong muscular movement. It feeds by individual cells having phagocytosis and absorption of organic matter. So basically, this is a lot smaller than those, but it makes its living in basically the same way. And many of us now think that if this is a simple phylogenetic tree of animals, that if sponges are here, here's the familiar groups. This is things like corals and sea anemones and jellyfish. These are bilaterians, things with a front, back, top, bottom, left, right. It's most of animal diversity. And we think that a lot of these Ediacaran organisms are in this part of the tree of life. So they're the first major radiation of macroscopic animals that precedes the radiations that gave rise to most of the things we're familiar with today. Now, by about 555 million years ago, we do get into those upper parts of the tree. This is a thing called Kimberella, found originally in Australia, but by the hundreds of specimens in the White Sea of Russia. And again, these things are a couple centimeters long. Clearly, they have an anterior and posterior side. They apparently moved on a muscular foot. They had viscera on top of it. We know that they could actually they had something of a rasping organ like the radula of modern mollusks and could actually rasp algae free from the surface. We know they moved because we find them preserved at the end of their trackways. So by this time, we start to see the kind of organisms that dominate biology today. I won't show it to you, but there's also evidence that the other major late blooming glutinidarians was around by this time as well. And then, as many of you know, by the time we get to the Cambrian period, we get what's commonly called the Cambrian explosion, where within about 25 million years, we see early representatives of almost all of the groups of animals that make fossils that will be found in the oceans for the rest of geologic history. So there are arthropods, there's a trilobite, other kinds, there are mollusks, there are annelid worms, there are actually fish by about 520 million years ago, all sorts of trackways and other trace fossils that tell us about both behavioral and functional diversity of animals. It's a real sea change in the composition of the biota. It turns out that once again, this biological series of events plays out in the context of major environmental changes. So going back to Tim Lyon's thing, he shows an abrupt increase in oxygen at about this time, and I don't think that's the case. This is from a paper by Xiaoming Liu et al. Again, two things again. Xiaoming did all the work and I'm an et al. But this is using cerium chemistry, and it suggests that, yes, around this time, oxygen started increasing toward its modern level, which it probably didn't reach until at least, say, 400 million years ago. But yes, oxygen is going up. Now, you can ask, well, how much oxygen do animals need? And there was a really interesting experiment done by a guy named Dan Mills in Don Canfield's lab about a decade ago, where they grew sponges at very, very low oxygen tensions. Somewhere between 0.5% and 4% of present-day oxygen. And in some ways, it's an overstatement to say that the sponges thrived, but they didn't die. And so, and there's a subtle difference between the two. So they argued that, well, you don't need much oxygen to make an animal. And I think that's true. I think many of us think that early animals probably didn't need anything like the levels of oxygen in the oceans today. But another of my old postdocs, Eric Sperling, now a professor at Stanford, did this wonderful meta-analysis of ecological surveys in the modern ocean along oxygen gradients. And it turns out that at the levels of oxygen that they used here, almost all animals in the oceans would die. So you don't need much oxygen to make simple animals. To make complex, energetically exciting animals, you probably do need more oxygen. And this is just from some of Eric's work showing that when he looked at things like polychaetes, because they're very diverse along these oxygen gradients, he found that in really low oxygen levels, there's no carnivores. But as oxygen goes up, you get more and more carnivores. And by the time you're at about 10% of today's oxygen levels, you have a pretty diverse fauna. And you might ask, well, why? Why should we worry about carnivores? Well, there's reason to believe that carnivores were ecologically important in spurring Cambrian diversification, because you have some animals specializing for different prey items learning to specialize for the avoidance of predation. And so in this case, you can't have that until you get oxygen at levels that are substantially above what most of us think was around in the Proterozoic eon. And this is just another diagram from a paper that Eric and I did. And again, it shows that as you start with zero oxygen, you don't have much diversity, you don't have much evenness, you don't have much carnivores. But again, by the time 8 to 10% of today's levels, you're doing okay. So you don't have to go up to modern levels to account for the diversity of animals that we see in the Cambrian explosion. But you do need more oxygen and potentially more food. And that brings up my last item, and that is that there is evidence that the food supply was changing. This is from a really nice paper, again, from my another one of my old postdocs, Yelk and Brox, in which they went carefully through rocks from before into the Ediacaran and into the Cambrian, looking at different steranes. And what they find is that before the Ediacaran period, there's very little in the way of steranes made by eukaryotic algae. 600 million years ago, there's one, and then by the time you start seeing Ediacaran animals, everywhere they look, primary production is dominated in many ways by green algae. And the fossil record of green algae actually corroborates that. I'm going to skip that one just so you have time. But now that's interesting to me. You know, why would you shift from dominance among primary producers by cyanobacteria for billions of years to ecological importance of eukaryotic algae? And it's interesting that if you go into the oceans today, where nutrient levels are low, you still have domination by cyanobacteria, mid-ocean gyres, for example. But where nutrient levels are higher, then you start getting eukaryotic algae. And so I at least find it interesting to think that what we see in space today may find a parallel in what we see in time 600 million years ago. And in that case, we'd have, you know, more larger algae contributing to more robust, more energy-rich food chains. And then finally, well, if that happened, why would it happen? And to me, once again, the answer has to do with phosphorus. Now, I first started working on phosphatic fossils in Ediacaran rocks years ago with this wonderful man standing at the base of that cliff of phosphate named Zhang Yun, who was Xu Hai's undergraduate and master's advisor. And this turns out, and Xu Hai did a lot of this work, to be one of the most remarkable fossil deposits in the whole Precambrian. You know, multicellular algae, all sorts of things which are at the very least related to holozoans, the sisters of animals, if not animals per se. And what's interesting is that the amount of phosphate in this deposit, about 600 million years old, is greater than all the phosphate in all the deposits on Earth deposited before that time. And this is not alone. There's phosphate, you know, large phosphate deposits start getting deposited widely about this time and continue to be deposited until the present day. So it seems that the world of phosphorus and therefore the world of nutrients is changing. And I'll just close by noting that there is another tectonic event, something called the Trans-African orogeny, and that yields what are thought to be the first Himalaya scale mountains, perhaps ever, but certainly for more than 1 billion years. That would increase weathering and erosive introduction of phosphate into seawater. And that's okay, because that could increase, you know, that could have some increase in phosphorus. But note that today, only a very small part of the phosphorus that is taken by primary producers in the oceans comes from, directly at least, from weathering and erosion. Most of it comes from an efficient remineralization and upwelling to bring it from the deep sea. So a thought that we had, it may be wrong, but I think it's at least worth suggesting, is that you increase phosphorus through tectonic means. That gives you more primary production, more oxygen, more oxidants, more efficient remineralization of organic matter, which increases the load of phosphorus coming back up. That increases primary production more, and you have a feedback cycle that can take you to another steady state. Okay, so the bottom line then is that our familiar world of animals may owe itself to tectonic events that spurred an increase in food and oxygen, which was a world in which animals could thrive. So, the punchline. The first, I hope I've convinced you that although imperfectly, we can reconstruct the deep history of Earth and life from the geological record, and that we have to think about physical and biological processes interacting and, you know, having an effect on each other through time. They're not independent actors. Major biological transitions, I think, both track and drive environmental changes in Earth history. And again, this point that just because I hang out with astrobiologists now and again, that what might make Earth unusual is not that life started here, but that it's persisted for 4 billion years. And that could actually be unusual. And with that, I'll just thank everybody who has helped me over the years, including a number of people in this room, and happy to take questions. Question, I have terrible eyesight, so Jay will run the questions.
Does this work? Yes, it does. Any questions?
Yeah, what's a catch box?
Thank you very much, Dr. Knoll. It's a wonderful lecture and I enjoyed it very much. I do have a question before I leave to keep on the thought from the chair. I was educated with believe that life on Earth was originated from the hydrothermal vents of the deep ocean. And actually, we go to the Museum of Natural History in DC. There's video tells us that life originated from the ocean. I recently read an article that says actually reported that thing, and then I read the original paper and said they found a lake in British Columbia and has a very rich concentration of phosphates, and then could potentially has the condition form molecules of like RNA, DNA, and ADP. If there's such lakes in there was such lakes in on the earlier, so I was just wondering. My question is, how confident are we about the theory of hypothermal vents?
I will say that there are people who are absolutely sure they're right that life began in a hydrothermal vent, and there are people who are absolutely sure that those people are idiots and that life began in a lake. And most of these people will shoot each other on sight. I think there is a case to be made, and a number of people like John Sutherland, who's a prominent origin of life chemist, has shown why lakes are good. Lakes enable you to concentrate materials. Lakes can, you know, evaporate and come back, and that has things that help you in origin of life. David Catling has written, I think, very wisely about this. So right now, I think, you know, and at least a number of people think that lakes in general, I mean, this new one up in British Columbia is really interesting, but lakes in general might be good. The loyal opposition, which is probably the nicest thing that has been said about these people, is that hydrothermal vents under the right conditions give you an energy source that could actually help you to find metabolism. And I must admit that when I listen to this debate, which I'm forced to do on a regular basis, I always say to these people, go to Iceland. There you have hydrothermal vents sitting beneath lakes. So it may not be this or that. I think there's a lot of room for thinking more carefully about the geologic circumstances. Sure.
Andy, I want to push you a little more on that. What is the partial pressure of oxygen in the benthos of the ocean? If you're really down in the abyss, how much oxygen is there?
It depends where you are spatially. It can go down to zero, and it can, in shallow waters, can be essentially saturated with respect to modern levels of oxygen.
Does this affect at all how we would interpret the biogeochemistry of deep sea vents?
that's a very interesting question, and I'd have to say I don't know, but when I read these papers, I think they still count on these being largely aerobic environments at the surface, and then having a gradient that goes down to sulfidic environments at depth. So yeah, oxygen is not, I think it's not irrelevant, but I'd have to do some homework to know exactly what they think is the importance.
We're a curious table over here.
Yeah.
I'm not an expert in GOE, but I had sort of always imagined that it was sufficient, that its timing was sufficient to be explained by just the large amount of iron that was in the ocean and in the Earth's crust, and that the GOE just starts when we run out of iron to stop being oxidized. But it sounds like that's maybe not sufficient. Can you say a little more about that?
Well, no, I think it's not irrelevant, and you're right for bringing it up. I think in the days of, one of my teachers, the late Dick Holland, he would have thought, because Dick was not prone to think that biology was very important in the history of environments, and so that he, I think, would have thought that, mantle fugacity might be changing, so the supply of ferrous iron might be running out. What doesn't work about that is the increasing amount of observations that suggest that the deep oceans remained ferruginous, that is iron containing, probably up until the Devonian. So it wasn't like we ran out of iron. There might be less, because again, with a cooling mantle, you should, according to people like Lee Kump, get lower fluxes of ferrous iron from ridges. So yeah, I think it's a fair criticism that one might, that might be another important factor. But I think I just think it's very unlikely to be a coincidence that right after we see large emergent continents, we have a GOE. That's a good point.
Hi, thank you for the great talk. I was just wondering about the environment of the pre-GOE. In a modern environment, archaea are mostly extremophiles, so relegated to sort of thermophilic or halophilic environment. Do you think that the environment of that era would yield more archaea, more prevalent in the environment, or do you think it was mostly bacteria?
Yeah, a couple of things to that. One, archaea have this bad habit of not showing up in the geologic record very well. We do in late Archaea and earliest Proterozoic, particularly lake sediments, see carbon isotopic fractionation that's suggestive of methanogenesis followed by methylotrophy to keep isotopically light carbon in the system. But again, I think textbooks mostly tell you that archaea are largely extremophiles, but in fact, there's been a large and increasing number of genomic projects of sampling the environment, and archaea are all over the place. They are subordinate in numbers to bacteria in most places, but it's not like they're absent. So I think that they would have played a role. In a world with low amounts of oxidants, so there's not much in the way of respiration, then methanogenesis. Today you see methanogenesis usually when you run out of oxidants for respiration, but it could have been proportionally more important in the Archean. So yeah, I think archaea were probably important members of the biosphere through the Archean.
Since cyanobacteria were clearly around before the GOE, why do we not see these localized iron deposits where the cyanobacteria grew more abundantly and may have caused iron to potentially cause solution in the fossil records?
Well, we see a fair amount of it because much of the iron in Archean iron formations is actually hematite, so it is iron oxides. Now, it turns out you can oxidize iron by having iron-based photosynthesis, discovered by Fritz Whittle around 1990, something like this. So you do get, the oxidation of ferrous iron in solution does lead to iron hydroxides. Also, there are photochemical processes that can do that, although whether they can do it on the scale that's needed to display the record, we don't know. So there are a variety of ways of oxidizing iron that don't even necessarily involve O2. That said, when I was a kid, I think most people thought that oxide iron formation did reflect cyanobacterial photosynthesis. That fell out of favor for a while, but one senses that it might be coming back. So it may be that cyanobacteria were more important than we thought, and at least in part from relevant to the question asked a minute ago, that they simply weren't producing enough oxygen to get rid of all the reducing materials in rocks and dissolved in seawater.
So one last question, because we have to clear out before 5, or around 5:20. We'll try to get to it.
Cecilia.
Thanks. Yeah, awesome overview of the history of everything. I was wondering, so in the fossil record, as you transition from single-cell organisms to the record of single-cell organisms to the record of multicellular organisms, I have always been a little bit puzzled about how we distinguish between like consortia of symbiotic microbes and true multicellular organisms. And whether or not it's even possible to do so looking at microfossil morphologies or subtle variations in the carbohydrate composition of the keratin and things like that. I haven't seen this new paper on the oldest multicellular organisms, so maybe this speaks to that. But yeah, I wonder if you could talk a little bit about that.
Sure. And Cecilia's heard all this before as an undergraduate, so she could ask some good questions. Just colonies are groups of cells that are found together where the unit of selection is still the individual cell. Each of those cells is capable of dividing, capable of going its own way. They don't particularly trade nutrients and that. So those are colonies. When you have a group of cells that sticks together in which there is sharing of nutrients, not all cells become reproductive cells, and so the unit of selection becomes the multicellular entity, then that's a true multicellular organism. I think the way we can at least tell sometimes when we've reached that stage is, some of the things we see in the Mesozoic, they're like blades of seaweeds and things like that. That's not likely to be a colony. That said, there are people who have been arguing for multicellular eukaryotes back at 2100 million years old, and that stuff, well, a lot of it actually looks like pyrite crystal structures, but those that look biological could easily be not only colonies, but bacterial colonies rather than eukaryotic. Now, and I should say, when I talk about complex multicellularity, what I mean by that, it's sort of an idiosyncratic definition, but it seems to be fairly widely held now, is that a complex eukaryotic organism is an organism in which tissues and origins circumvent the limits of diffusion. And so a circulatory system, if you were dependent entirely on diffusion, you could not be three-dimensional, basically. And so animals, that hasn't happened much. There's 40 of the 120 different major clades of eukaryotes, nearly half of them have simple multicellular organisms. Only 6 groups have complex multicellularity.
Thank you again for this excellent talk. Wow, it's very exciting. I'm going to try to make the question a little bit briefer, but it appears that there's evidence that there's a sort of microbial intelligence, right? Either from like bacterial reorganization under antibiotic stress, or myzobacteria coordination, and cooperation, and some of the things you had mentioned as well. My question is, do we have any kind of archaeological evidence that this same sort of microbial intelligence one cell being able to maneuver and reorganize or cooperate existed way back in the beginning, before the GOE and everything. Do we have sort of evidence of that?
Yeah, there was actually a book reviewed in Science magazine about 2 weeks ago that actually argues for such a concept of microbial intelligence. There's certainly microbial behavior. That is, organisms respond to cues, either from the environment for other organisms. I would probably stop short of calling that intelligence in the same sense that we have it. But yeah, they're just in orientations, in the way different organisms are related to each other spatially, and that there's certainly evidence, I think, for behavior among microorganisms as far back as we can see in the geologic record.
What was the name of that book?
Oh, I can't tell you but if you look in Science over the last couple of weeks, there is a review of it, and you can judge for yourself what you think.
So let's all give Andy a